Biological classification. Aristotle was the earliest one to attempt to group the animals into class, like he divided the plants into trees, shrubs, and herbs. Then Linnaeus divided the animals into Plantae and Animalia, two kingdom system. So he did not distinguish between eukaryotes and prokaryotes, unicellular, multicellular, and photosynthetic and non-photosynthetic. After this, many different attempts were made, taking in considering many characters like gross morphological, like cell wall, mode of nutrition, habitat, etc. After this, R.H. Whittaker came up with the five kingdom system that were Monera, Protista, Fungi, Plantae, and Animalia. So, the question got like which characters were used by him. So it was cell wall, cell type, nuclear membrane, body organization, mode of nutrition, and reproduction of phylogenetical evidence. पण ह्याच्यामध्ये we know that a cell wall is not present in animals, whereas Monera cell wall is non-cellulosic. Protista मध्ये it is cellulosic. Fungi मध्ये it is made up of chitin, and plant it is made up of cellulose. After this, the Monera आपण further two group मध्ये divide केलं, like Archaebacteria, Eubacteria. This is a three-domain system. Three-domain system में Monera is further divided into two another groups. Earlier, bacteria, blue-green algae, fungi, moss, ferns, gymnosperm, and angiosperm were grouped under plants, and other prokaryotic were grouped with the eukaryotic. So this was wrong. So R.H. Whittaker survey was the most accepted way for grouping animals. After this, kingdom Monera. These are most abundant microorganisms. Coccus is spherical in shape. Bacilli are rod shape. Spiralia are spiral in shape, whereas Vibrio are comma in shaped. So here thing to remember, most important thing is they show very complex behavior and extensive metabolic diversity, though their structure is very simple. Question alignment almost 10 to 12 times that which of the following shows extensive metabolic diversity. We usually tend to mark Animalia, but it is Monera. They have a very complex behavior. So terminology they activate obligate is strictly, and facultative is like they need, but it's not the necessity. So here Archaebacteria. So these are found in extreme conditions because they have some difference in their cell wall structure, that is peptidoglycan layer present असते. Salty areas, they are known as halophiles. Hot springs, they are known as thermoacidophiles. Marshy area, they are known as methanogens. Methanogens are also present in the guts of several ruminants, which help in the production of methane gas. Eubacteria, the true bacteria, they are characterized by presence of a rigid cell wall. If motile, they have a flagella. Cyanobacteria, also known as blue-green algae, have chlorophyll A similar to the green plants and photosynthetic autotrophs. These are Monera. They have chlorophyll similar to the plants, that is chlorophyll A. A cyanobacteria unicellular, colonial, or filamentous, freshwater, marine, or terrestrial algae. So the colonial form, it is a nostoc. This have a gelatinous sheath around them, and this is a heterocyst which helps in nitrogen fixation. But heterocyst is an obligate anaerobe, so its function is disrupted when it comes in contact with oxygen. So here we have a chemosynthetic autotrophic bacteria which oxidizes various inorganic substances and use their energy for the ATP production. These are chemosynthetic autotrophs, whereas heterotrophic bacteria are completely dependent on other animals for their nutrition. Mycoplasma are the organisms in living cells that can survive without oxygen. These are the smallest living cells. Many mycoplasma are pathogenic. They lack cell wall. Kingdom Protista. Protista is further divided into following groups. First is chrysophycetes. Chrysophycetes include golden algae or desmids and diatoms. These are found in both freshwater and marine water. They float passively. Their structure is overlapped like a soap box and they are indestructible. They are known as diatomaceous earth because when they are dyed, their skeleton or the covering it gets accumulated at the base surface of the water body. It is used for polishing filtration of oil and syrups. Diatoms are the chief producers in the ocean. So things to remember here. Then the structure is soap box. They are the chief producers. So if question is asked a chief producer कौन है, we always tend to mark algae. And in this case the chief producer is chrysophytes. Example are diatoms and golden algae. After this, dinoflagellates. Dinoflagellates are usually marine or they may be photosynthetic. They have a stiff cellulosic plates on the outer surface and most of them have two flagella. One longitudinal and one is transverse. One is longitudinal, this one is transverse. So the important feature to remember here is they have a stiff cellulosic plate and the transfer is lies in the furrow between the walls plate. Very often red dinoflagellates undergo rapid multiplication and they make the sea appear red. Toxins released by such large number of dinoflagellate population may even kill other marine animals such as fishes. Things to remember: they have a two flagella. One is latitudinal, one is transverse. They have a stiff cellulosic plate and they multiply too fast. Red algae, for example, Guanolax, which is responsible for red tides. Usually if we are asked the red tides, then we can like red algae. No, it's because of dinoflagellates. Euglenoids, things to remember: it has a pellicle layer which makes it flexible. They are photosynthetic in presence of sunlight. An example is Euglena. Slime mold, they are saprophytic. Segregation is known as plasmodium. They form fruiting bodies bearing spores in unfavorable climates. These have true walls. Spores have true walls. In protozoa, amoeboid, that is, they live in fresh water and seawater. After this, they prey by putting their pseudopodia and they have silicaceous shells if they are marine. Example is entamoeba. After this, flagellated is trypanosoma, sleeping sickness, and they are parasitic and have flagella. Ciliated is paramecium. Cilia causes water ladder movement through a body cavity known as gullet. And sporozoa, example is plasmodium, whereas in that piche wala thing the aggregation is known as plasmodium. Here the example is plasmodium. It is malaria parasite and they are spore-like stage in life. Kingdom fungi. Kingdom fungi maximum are multicellular, whereas yeast is unicellular. Rust causing is Puccinia, antibiotic is penicillium. So here yeast is unicellular. Their body consists of long slender thread-like structure known as hyphae. So if hyphae is like continuous tube filled with multinucleated cytoplasm, it is known as synocytic hyphae, and if it has branches or septae or cross wall, it is known as septate. The cell wall is made up of chitin and polysaccharides. Most fungi are saprophytes, parasites. Lichen and fungi in association with algae as lichen or with the roots of the mycorrhiza. Fungi act as symbionts. Reproduction, so first is plasmogamy, that is fusion of protoplasm, then fusion of two nuclei, karyogamy, and then meiosis in zygote. Sometimes the karyogamy is at a halt, that is dikaryon phase. It has n plus n nuclei. It occurs in Ascomycetes and Basidiomycetes. So Phycomycetes, things to remember, it is aseptate, that is cenocytic. Zoo and the asexual spores are zoospores, they are motile, or aplanospores that are amotile, non-motile. These are endogenously produced in a sporangium. Zygospore is formed by the fusion of two gametes. These can be similar, that is iso, or dissimilar, aniso and oogamous. Examples: Mucor rhizopus albugo, that is parasitic fungi on mustard. These examples are very important. Mucor rhizopus and albugo. Ascomycetes, sexual way is absent here. Ascomycetes are commonly known as sac fungi. Penicillium and yeast, that is Saccharomyces, are example of this. They are saprophytic, decomposer, or coprophilous, that is growing on dung. After this, त्यांचा वॉल असतो ब्रांच अँड सबटेट. आगे ह्यांचा पण ब्रांच अँड सबटेट वॉल असतो. याच्यामध्ये कोनिडिओफोर्स आर एक्झोजिनियसली प्रोड्यूस्ड, whereas ascospores are endogenously produced in a sac-like structure known as ascus. Example is Aspergillus, Claviceps, Neurospora, Morels, and Truffles. Neurospora is known as Drosophila of plant kingdom because many experiments are carried on it. Basidiomycetes, these are known as mushroom bracts or puffballs. But Morels and Truffles are mushroom, but these are the example of Ascomycetes. Asexual spores are not found. Ascoमध्ये both, Phycoमध्ये only asexual, and whereas in this asexual spores are not found. Fragmentation very common. A dikaryotic stage is present which gives rise to basidium, which on development produces 4 basidiospores. And meiosis takes place, important point, meiosis takes place in basidium producing 4 basidiospores. These are exogenously produced. Example is agaricus mushroom, ostilgo, that is mud, and Puccinia, that is rust. Deuteromycetes is imperfect fungi because only asexual form is known. These are produced only by asexual spores like Conidia. Conidia is exogenously produced, and septate and branched asta hyphae. Example is Elytratria, Coletrichum, and Trichoderma. Viruses are non-living or not truly living creatures. These are inert crystalline structure around outside the host. They are obligate parasites. So here the virus name means venom or poison was given by Dmitry Ivansky, and he found these virus from tobacco mosaic disease. Then M.W. Bejanek, he extracted the infected plant and he gave the name Congetium vivium fluidum. So virus was termed by Dmitry Ivansky, and the it was isolated from infected plant by M.W. Bejanek, and it was termed as Contagium vivium fluidum, that is infectious living fluid. W.M. Stanley crystallized this structure, and it found to be like proteins and RNA. They are obligate parasites. So it has a DNA, that is double stranded DNA or single stranded DNA. A single stranded DNA is usually not present because DNA is complementary, so it either has double stranded DNA, double stranded RNA, or single stranded RNA. So, which infect animals are either single or double stranded RNA or double stranded RNA. But whereas which affect plants are single stranded RNA. Plant ssRNA, but animal it can be ssRNA, ssDNA, or dsRNA, all type. And then these can be it has a outer structure made up of units of capsomer which are helically or polyhedrally arranged. Examples of diseases causing are these AIDS and mumps वगैरह वगैरह. After this, viroids are just free RNA, not protein. So virus has protein and RNA. Only RNA, viroids. Only folded protein, prions. So viroids were discovered by T.O. Diner, and prions causes disease such as in cattle. It is known as BSC, bovine spongiform encephalopathy, which is known as mad cow disease in cattle, and CR-Jacob disease in humans. After this, lichen, algal component known as phycobind, fungal is known as mycobind. They do not grow in polluted areas. So, question usually asked that where do you like if a place is found to be polluted, which of these will not grow? So it is lichens, they do not grow, they are good indicators of pollution.